Today we are going to talk about process of fertilization, right? First of all, we have to define what is meant by fertilization. Who will define fertilization for me? Yes. And this is something every medical student knows. Somehow everyone is curious about it. Yeah, who will define fertilization? Okay, he has made a very simple definition. The simplest definition of fertilization is the process of fusion of male and female gametes, right? What is fertilization? The simplest definition of fertilization is the process of fusion of male and female gamete, right? That is process of fusion of sperm and ovum. Is that right? This is the simplest definition, but you know, in your life sometimes you come across very fussy people and they want a very good definition and very precise definition. For those people, write another definition. We tell the same process more clearly. Fertilization is a coordinated molecular process, molecular and cellular process. Fertilization is a coordinated sequence of molecular and cellular process which begins with the contact of oocyte and sperm. So fertilization begins with the contact of oocyte and sperm and ends with the intermingling of maternal and paternal chromosomes, and process ends with the intermingling of maternal and paternal chromosomes at metaphase of first mitotic division. At the metaphase of first mitotic division of the zygote. Do you think zygote is an embryo or not? Yes, anyone good in embryology? Zygote is embryo or not? It is not embryo. Okay, anyone who says zygote is embryo, raise your hand. Anyone who thinks zygote is an embryo? No one wants to be right. Write it down. Zygote is a unicellular embryo. Oh my God, so sad, right? Zygote is a unicellular embryo. Embryo is the early development of the product of conception, isn't it? So development of product of conception starts from zygote. So zygote is an embryo which consists of only single cell. Is that right? Okay, let me repeat. What is fertilization? The simplest definition is fusion of male and female gamete. Is that right? This is fertilization in simplest way. Or fertilization is a molecular and cellular process in which begins with the contact between the oocyte and sperm, and this process ends with the intermingling of maternal and paternal chromosomes at the metaphase of first mitotic division of zygote. That is the beginning and the end of fertilization process, right? Okay. Now, what is the most common site of fertilization in human being? Which is the most? Ampulla of the fallopian tube or ampulla of the uterine tube. There are ampullas of many other things in the body, right? So it's the ampulla of the fallopian tube or uterine tube. What are the different parts of fallopian tube? What is this part called? This part? No, these processes are called Fimbria, but this part of it is not called ampulla. The first part is infundibulum. Then here is ampulla. Ampulla is the widest and the longest part. But earliest part, this one, is infundibulum. Infundibulum is catching the ovum. Is that right? And then ovum moves in the ampulla. And ampulla of the uterine tube is the most common site where most commonly you can say fertilization occurs. And then this is ampulla, and then this is isthmus, and then intrauterine part of the fallopian tube. Is that right? Now, again, what we were talking about was that most common site for the fertilization in human beings is, yes, ampulla, and ampulla is having these finger-like processes which are called fimbria. Is that right? Now, for example, let us suppose that fertilization is going to be at this site, okay, at this point. It means ovum has to reach here, has to be transported here, and sperms are deposited where? In the uterus or in vagina? During ejaculation, semen is deposited in the vagina, right? So sperms are deposited over here. This is the semen, right? And sperms are present within the semen, isn't it? Now, sperm has to have to travel through the cervix, through the uterine cavity, through the fallopian tube up to this. It means that for fertilization process to occur, male and female gametes should reach up to ampulla. So ovum should be transported after the ovulation when ovum is released, right? During the process of ovulation, when ovum is released, it should be transported to the ampullary region, and if fertilization is going to be there, then of course sperm should also be transported from the upper part of vagina through the cervix, through the uterine tube, through the isthmus, and then until ampulla, and there they should fuse. So really, before we really start talking about how the fusion process occurs, we should talk about how ovum is transported up to this point and how sperm is transported from here to here. First we talk about the ovum. You know, ovum travels less and sperm has to travel more. You can understand it well. It's a very feminine feature. Women like to travel less and weight more, and men get almost wild to travel as long as they can. So sperm have some manly feature and ovum has some feminine feature, right? So ovum travels less and sperms travel a lot, right? Ovum is motile cell or immotile cell? It is immotile. It is not motile on its own. It doesn't have any type of cilia or tails or anything, right? Ovum is immotile. Sperm is of course highly motile. Healthy sperm has a tail, and that really propels the ovum through, propels the sperm through all this process. Is that right? Now, first we will talk about transport of the ovum. Then we will discuss about the transport of the sperm, because transport of ovum is very few things to talk about. First of all, we will say that at the time of ovulation, right, mature Graafian follicle ruptures and ovum with some fluid, ovum with surrounding follicular cells with some fluid is expelled out on the surface of the ovary at the time of ovulation. At the time of ovulation, there is a sudden increased level of FSH and LH, especially there is a dramatic increase in the luteinizing hormone level at the time of ovulation, just before few hours of ovulation, right? As soon as ovum come out with the follicular cells, and these follicular cells, right, they rearrange around the ovum, and these rearranged cells are called cells of corona radiata. These are called cells of corona radiata. So ovum comes out with the fluid, and this ovum has zona pellucida around it, which is the glycoprotein cover, and it has a few follicular cells which are now called corona radiata cells. Is that right? They are released on the surface of the ovary. Now, around the time of ovulation, the fimbria of the infundibulum become really very active. Even just before the ovulation, due to hormonal activity in the body, fallopian tubes, infundibulum having the fimbria, these fimbria start sweeping on the ovary. I don't know, I cannot really praise the nature for all these coordinated things, that it is energy saving, I think, that whole the month fimbria are not so active, but just around the ovulation fimbria become active, right, and they start sweeping on ovary, and where they found a little bulge, they become more active around that. They are hoping from there what will come out? Ovum will come out. Is that right? And this movement of the fimbria is hormonally controlled. Role of luteinizing hormone, estrogen, right, and other hormonal factors is playing into that. Now, as I told you, that ovum is immotile. It does not have any motility activity. So it has to be picked up by the fallopian tube. It has to be picked up, otherwise it will be lost into peritoneal cavity. So there are two processes by which it is picked up. One is the sweeping movement of the fimbria of fallopian tube on the surface of the ovary. This is the one mechanism. Secondly, within the fimbria, right, there are, there are lining of the mucosal cells. Fallopian tube has mucosa. Those mucosal cells are ciliated. Right, fallopian tube has mucosa and mucosal cells are ciliated. So fimbria, suppose these are the fimbria. My fingers are fimbria. Now they are having the mucosal cells here, and these mucosal cells are ciliated. When fimbria are sweeping, additionally, those cilia are also moving and producing small fluid currents. Small fluid currents. So those fluid currents also help the fimbria to pick up the ovum. Is that right? So how is the transport of the ovum? That at the time of ovulation, we say that sweeping actions of fimbria and fluid currents are also produced by the cilia of the mucosal cells of the fallopian tube. Is that right? And all these sweeping actions and fluid currents help the fimbria to pick up the ovum and take it into fallopian tube. Is that right? Now, what is this ovum? Is it definitive ovum? It is primary ovum, secondary ovum, or definitive ovum? I'm about to be impressed by someone. There are three types of ovum. A ovum rather passes through multiple stages. But which ovum is picked up at the time of ovulation? Or we can say which ovum is released at the time of ovulation? Is it primary ovum? Or it is secondary ovum? Or is it a definitive ovum? It is not definitive. This is not definitive. Because definitive ovum is produced when magic touch of sperm come to the ovum. Then ovum in hurry complete its second meiotic division. Is that right? So it is secondary ovum. This is not definitive ovum. Definitive ovum is produced during the fertilization process. If no sperm come, secondary ovum never converts into definitive ovum, right? So what is this? This is secondary ovum, which is passing through the process of second meiotic division. At the time of ovulation, ovum has completed its first meiotic division and it is passing through the second meiotic division. If sperm comes and touches it, it will complete its second meiotic division and convert into definitive ovum by releasing one more polar body. But if sperm never comes, secondary ovum will move a little bit. And how much it survives? What is the normal life of secondary ovum? 1 to 2 days. In most females, only 1 day. Only 24 hours. Again, this is a feminine behavior, not waiting long. As female reproductive total years are less and male reproductive years are more, in the same way survival of ovum is less and sperm is more. Ovum wait usually 1 to 2 days, more accurately around 24 hours it will wait, is there sperm around or not. If there is no sperm around, this ovum, secondary ovum, will never complete it second meiotic division, and without completing that, it will die. Is that right? And it will be reabsorbed by the female genital tract mucosa. Is that right? Or neutrophil macrophage will come and eat it up and finish its story, right? And what is the life of sperm? Okay, they survive at least for 48 hours. Is that right? But they, in some situations, they survive for many days. We'll talk about that later. Now, so ovum will reach here and survive for about 24 hours. Meanwhile, it may be traveling towards the uterine cavity, right? But if fertilization is going to be there, most commonly it occurs here. Is that right? Now, we have learned that how ovum is picked up from the surface of the ovary into the fallopian tube. Then it moves a little into fallopian tube. What is the mechanism of movement of ovum or mechanism of transport of ovum within the uterine tube? Your uterine tube and fallopian tube is same. Yes. No, that is not that good answer. Ciliary movement do not play a major role. Major role is by the peristaltic contractions, peristalsis of the fallopian tube. Peristalsis means contracting and relaxing movement. right? So actually, there's a little peristaltic movements which transport the ovum through it. Is that right? So picked up is by the Fimbria movement. Is that right? And peristaltic movement moves the ovum through the fallopian tube. Am I clear? Okay, this was a little travel. Let's talk about this happy man who is deposited here. How it travels such a long distance. Is that right? Now, this man has a tough competition. Normally, on average, how many sperms are deposited in one ejaculate? Yeah. Pardon? Couple of 100 millions. You can be a little more specific. It just reminds me of Bill Gates. He is billionaire. Yeah. Not millionaire. As far as sperms are concerned, men should be happy they are millionaire. Yeah.
Not all men.
All right, but at least they do make their wife millionaire as far as sperms are concerned. Am I right? So, it is approximately 200 to 500 million sperms. 200 to, look at the generosity of men. 200 to 500 million sperms are usually deposited during one ejaculate. Is that right? And in the semen, sperm make how much component of the semen? Semen is the male, fluid which is released in the female reproductive tract, right, in the vagina. This usually it is 2 ml to 6 ml, varying a person to person and occasion to occasion. But usually, for example, if you have 1 ml semen, how much percentage of that will be made by the sperms? About 10%. 90% of the different fluids coming from prostate gland and seminal glands and other glands. Is that right? 10% of the mass of the semen is made by the sperms. So sperms are deposited here, and there's very important enzyme coming here. What that enzyme is doing? That coagulates some of the proteins here. There's an enzyme coming along with the semen. Within the semen, there's an enzyme which is activated here, and that helps some part of the semen to be coagulated so that it does not fall out, rather it does not drains out, right? Some of it remains sticking to the cervix. What is that enzyme? Function of the enzyme is to coagulate, so it has coagulase activity. But because that enzyme is released by the seminal vesicles, so that is called vesiculase enzyme, right? So sperms which are deposited here, right, and vesiculase enzyme is activated, which produces a small coagulum here, so that some of the sperms at least remain here for longer time. Is that right? And then sperms are motile. Okay, a very interesting question. Sperms are more motile in vagina, or they are more motile in uterine cavity? They are more motile in vagina. Okay, anyone else? This, according to her, sperms are more motile in vagina. Anyone has a different idea, or everyone agrees with her?
They get tired.
Don't give me reason, because you're wrong.
I'm wrong?
Yeah.
Oh, I thought
Look, vagina is an acidic environment, and in acidic environment, usually sperm should be deposited into adult female. Is that right? And adult female vagina is acidic. You know it. Acidic environment, lactobacillus are there. And in the acidic environment, right, sperms slow down. Actually, they become really super active as soon as they go into warm uterus, because uterus has alkaline environment, less hostile environment. Why vagina is acidic? Because acidic environment helps the vagina to protect itself from many microbes. But a little cost of that is it slows down the sperms a little bit. But sperms are very clever. They rapidly jump from vagina, they move into cervix. Is that right? But once they are in the uterine cavity, sperms become more fast, because environment in the uterine cavity is alkaline. Is that right? Another good news is that especially around the time of ovulation, when ovum is about to be here, the glands present in cervix, cervix has glands which produces mucoid material. There's a mucus in the cervix here. This mucus especially becomes thin, stringy, less viscid, and more abundant around the time of ovulation. It becomes thin, not thick. It becomes stringy. It changes its characteristic. You may be thinking, why Dr. Najib is so fussy about the small point that around the time of ovulation, this becomes thin, stringy, and it favors the sperm movement travel. Why I'm fussy about that? Because when you will go into high classes, you will study gyneology. One way to know that female has ovulated or not is check the cervix here. Doctors take little bit mucus from the cervix, put it on the slide. If it is thin, stringy, and making a furling pattern, they say she has ovulated. Is that right? Anyway, we will not go into detail. But this is good for the sperm, that mucus plug which is present in the cervix canal, that become thin and less viscid and more copious, abundant, stringy, and all these things favor the movement of the sperm. So movement pass through the, sperms pass through the cervical canal. What propels the sperm through this? Yeah, the tail of the sperm. Tail of the sperm moves, and these are the propulsion by the tail of the sperm, which help the sperms to travel from the vagina and through the cervical canal into, what is this? Uterine cavity. Is that right? Another thing. Okay, tell me. To move the, to produce such a movement, they have to move their tail, and then they have to go a long way. They need a lot of energy. From where the energy comes? No, but look, answer is right. One person says mitochondria, he's right. One person says ATP, of course he's right, because mitochondria will make ATP. But I'm saying what material they will take into mitochondria and make eventually ATP. No, it is not glucose, it is not sucrose, it is fructose. It is fructose. Yes, semen is very fruity. It has fructose. Good news not for you, it is for sperms. They take up the fructose, and then from there they produce energy. With that way, fruity thing, they move on the way, right? Now, hard workers, really. Even molecular levels, females behave differently than males, isn't it? And I'm so happy that males don't need to behave like females. I don't like to be pregnant. Now, so sperms, they will from where the fructose will come? From where the fructose will come? Semen will bring its fructose with itself, right? Semen is rich in fructose. Is that right? So sperm will take energy from the fructose, and someone was talking about the mitochondria. Where are the mitochondria in the sperm? In the head. You mean it that sperm has, let's suppose your theory is this is the happy sperm, and here are the mitochondria, and mitochondria act as a bumper and it will hit the ovum. Oh my God, this is not a news. Yeah, where are the mitochondria? Good medical. In the tail. I don't know, you are wrong. You say head, you are wrong. You say tail in the wrong, it means there is something else. Middle piece. There is something called middle piece here, Did you know all the mitochondria of the spermatozoa come together and fuse and make middle piece? So actually, what is middle piece? If you look in the head, there is no mitochondria there. Where the mitochondria have gone? All the mitochondria during the process of formation of the sperm, they fuse together and make the middle piece, and this is generating continuously energy for the movement of the sperm. Is that right? Am I really clear? Okay, so sperm is moving through that. While sperm is moving through this area, I must mention one thing. There are some mucosal folds in this cervical region, cervix. And in those mucosal fold, some of the sperms hide and keep sitting there and then keep on coming into action later on. That is why sometimes sperms, after many days, they are still moving around. That some of the sperm, they stick or they are held into mucosal fold of the cervical canal, right? And from there they may be gradually released. So it's wonderful. Some of them go straight away, and some of them are sitting there, and then gradually, as they feel better, they come out into action. Is that right? Right, one question I want to ask. First sperm will take how much time to reach from here up to here? What is the shortest time possible? Don't tell me before ejaculation.
6 hours.
6 hours. You are underestimating them. They are too fast.
4 hours?
They are still fast.
Faster than that?
5 minutes.
Wow.
Yeah, you should learn to appreciate the sperm.
It's like a mile.
It takes it so fast. I'm talking about the fastest, and they keep on reaching. The first one sometimes takes as early as 5 minutes after ejaculation. They have recovered some, I think, very naughty sperms present over here. So fast. But some of them may take up to 45 minutes. Right? So, but of course, within 45 minutes, all of them do not reach there. Right? Some of them are wandering around. Some of them, rather most of them never reach there. Out of 100, what do you think, out of 500 millions, how many reach here? Okay. Out of 500 million sperms deposited here, how much truly reach at the site of fertilization? Of course, for fertilization, you need only one. But they are coming like troops. Right? So how many troops reach here? Out of 500 millions, about 500, 200 to 500 only. It means any sperm which is reaching here is 1 in a million. You have heard the song 1 in a million, you are 1 in a million. That could be said about the sperm also. That any sperm which reaches here, we can sing you are 1 in a million. Isn't it? Is that right? So you have to remember, out of 500 millions or 200 millions sperms deposited here, maybe just 200 reach here. Where the others have gone? Don't tell me that they have gone through other fallopian tube to somewhere else. Where they have gone? Yeah? Yeah, they disintegrate on the way, casualties on the way. Many of them, they disintegrate on the way and they are absorbed by the female genital tract mucosa. Is that right? Am I?
They know which fallopian tube to go into, right?
No, actually, look, life is a lot about chances. They go to both sides. Some go here, some go there.
Not to the one side or no?
No. It is not like that. There's no red light there that you don't go here and green light for this area. No. Actually, some sperms go in that direction, others go to this direction. They only go under one condition to one fallopian tube. What is that condition? When there's only one fallopian tube. In many females, we remove one fallopian tube. Isn't it? Due to some complications. Otherwise, they love to go into both. So some troops go into this way and other troops go to this way. And someone, they have to find eventually where is the ovum. Is that right? Clear? Because they don't know, while they're moving here, they don't know where ovum is present on which side. Even many times, ovum is not there. Right? Anyway. So, once they have moved here, once they're passing through this cavity and through this area, contractions of uterine smooth muscles and fallopian tube smooth muscles also help. So, it's a double action. Mutual interaction between male and female for the mutual benefits, that sperm is contributing its motile motility and female is contributing by contractility of its hollow viscous uterine and fallopian tube, right? Both these things promote and facilitate the movement of the passage of the or travel of the sperm. Sperms are a bit intelligent, They bring with themselves in the semen prostaglandins. In the semen, not only there is fructose, there is also prostaglandins. So some prostaglandins which are moving up, they also increase or stimulate the contractions of the uterine tube. Is that right? Is that right? So these were something about the movement of the sperm, how sperms are passing through. Important point to remember that in the vagina there is acidic fluid, they move slowly. In the uterine cavity and fallopian tube, they become fast. Number two, another important, very important point. Some of the few lucky which reach here, what happened to them? Most of them who reach here, of course, a small percentage reaches. Once they reach here, they stop there. Now, fertilization occurs here, but most of them stop there. It means there will be something which will restart them and then they will move there. Who will restart them? Yes. Especially if ovum comes, of course it is having some follicular cells around it which are called corona radiata cells. If it really comes, they release ovum plus its friends. Its friends are corona radiata cells. All of them release chemoattractants like a good scent attracting the sperms, please come. So what happens? Ovum itself does not move much. It sits here and then ovum plus its corona radiata cells, they are releasing chemical substances which are chemoattractant. And once they are released, if ovum is there, they can sense the presence of those substances and then rapidly restart the movement and approach to the site of fertilization. Is it clear? Again, movement of the sperm by multiple things which facilitate the movement of sperms. Number one was its own tail movement. Number two, contractions. And this little favor also done by the cilia of the what? Mucosal cells, right? They also assist in the movement of sperm, right? But they slow down at the uterine part of, or slow down at the isthmus. And if ovulation occur, and product of ovulation, that is secondary oocyte along with its which cells? Corona radiata cells, they release chemoattractive substances and they become activated and they rush towards the ovum. Is that right? Now, before the fertilization really occurs, I mean before the ovum and the sperm really fuse, sperm has to undergo two important changes. Is that right? One process is called capacitation reaction, and other process is called acrosome reaction. We'll discuss both of them and you must know the difference of them. Sperm have to pass through capacitation reaction and acrosomal reaction before sperm is capable of fusing with the ovum. Is that right? Now what is capacitation reaction? Capacitation is special type of conditioning of sperm within the female reproductive tract, and it takes about 7 hours. Even the fastest sperm may take 5 minutes to reach here, but it cannot fertilize immediately. Is that right? What you remember was 7 hours, that was not the travel time, that is the capacitation time. What is capacitation? Capacitation is, capacitation is special type of conditioning of the sperm when it is present within the female reproductive tract. What is that special change? Let me tell you. Let's suppose here is sperm. Right? It's moving on the way. Actually, number one, you have to remember, on the head side, it has a double membrane. Do you know that? This is called plasma membrane or the cell membrane of the sperm, and there's a special membrane here which is called acrosomal membrane. It is a double membrane here. Then it has a lot of glycoproteins there. Glycoprotein deposited on its head. So it has to wash its head before it is greeted by the ovum. So head washing is called capacitation because its head is loaded with a lot of glycoproteins like substances. And until these glycoproteins are present over there, until they are not washed away, the sperm should be sad because it cannot fertilize the ovum. The dirty head. Isn't it? So who will wash its head? Yeah, of course. Now she owns the sperm, so she has to wash its head. Is that right? It's now your personal matter. So what really happens, that head of this will interact with the mucosal lining of the fallopian tube. And maybe uterine cavity and mucosal lining of the uterine cavity and fallopian tube, and there are enzymes from there, and cilia which wash its head, and enzymes digest away these glycoproteins. And once these glycoproteins are removed, right, now sperm is happy because, because now it is capacitated. It has developed the capacity to fertilize the ovum. Is that right? Now, why this point is important? And this capacitation process take about 7 hours in the female reproductive tract. Why it is so important to mention? Because when we try to do fertilization in vitro, in the laboratory, we put the ovum and put the freshly produced sperm on that. Freshly produced sperms, or rather specially released sperm in the semen, cannot fertilize the ovum. And doctors were really surprised how in the female genital tract or reproductive tract, sperms can fertilize the ovum, and when we put the both things in the petri dish, they don't work well. So later on, so they came to know that there's conditioning of the sperm, I think head conditioning, right, within the female reproductive tract. Is that right? And then they become really wise, and now they have some materials in the laboratory that freshly ejaculated semen, they put into that material, and that material will wash away the glycoproteins from the head of the acrosomal region of the sperm, and after that sperms can do successfully fertilization in vitro. Is that right? Am I clear? So this is one point, that it has to undergo capacitation reaction, right, and this occurs mostly in the uterine tube by the interaction between the sperm and the mucosal surface of the tube. These are glycoproteins. There are glycoproteins here, and plus some seminal plasma proteins, some seminal gland product, both of them may be there, glycoproteins or seminal plasma proteins. All of them need to be washed away or removed, right? Only capacitated sperms can pass through corona radiata. Sperms which are not capacitated cannot pass through the first barrier, that is corona radiata. After that, when sperm is capacitated, okay, we make a sperm at 2 stages. Right, first it is very, very sad sperm because it is not capacitated. Is that right? Then it is capacitated and it become a somewhat happy sperm. Is that right? Here is the ovum waiting. Okay, I should make a little bit bigger. Ovum, this is, and it has a special glycoprotein covering, and this glycoprotein covering is called zona pellucida, glycoprotein covering, and then around it, it is having its friends called corona radiata. Is that right? And corona radiata cells, they are held together by some connective tissue which is having hyaluronic acid. This is hyaluronic acid which is helping them to be held together, everything there. Right, so ovum is here and sperm is there. Right? Sperm is bigger or ovum is bigger? Ovum is bigger. It has a lot of cytoplasm in which it has even a lot of nutritional substances. Right? Now, once it is capacitated, its glycoprotein and seminal plasma proteins are removed, it will pass easily through that, right? But as soon as it touches the zona pellucida, it undergoes another reaction. As soon as it touches the zona pellucida, it undergoes another reaction. Let me show that reaction. This is a sperm, middle piece, tail. This is the membrane of the sperm. Is that right? This is its nucleus, which has 23 chromosomes, isn't it? And haploid number 1n number, and this is the acrosomal. Now, as soon as sperm touches the zona pellucida, right? Zona pellucida induces a reaction into this. That reaction is called acrosome reaction. Acrosome means acral end. I mean this is the end of the sperm, no? Head of the end. Now, during this process, acrosomal end, what really happens that inner membrane and this outer membrane, they fuse at multiple points. They undergo fusion at multiple points. They fuse. And when they fuse at multiple points, how it will look? Both membranes are fused here, and wherever they fuse, for example, this point and this point fuse, when they fuse with each other, they melt away. And apertures are produced, holes are produced, through which now it is doing its ultimate function. Oh, proud of himself. Right. What it is doing? It starts releasing enzymes so that it can really digest away the what? Zona pellucida. Female likes to put the barriers. Yeah, that is true. So it has done its head washing, and now it is releasing some substance as soon as it touches the zona pellucida. And when this is released, what is this substance? These are different types of enzymes to digest away the zona pellucida. These enzymes are acrosin, yes, acrosin, or some trypsin-like substance, or hyaluronidases, an enzyme, hyaluronidases. All these enzymes work together and help in digestion of what? Zona pellucida. Is that right? So actually, during this process, the stupid sperm will lose its head surface. You know? Now it wants to fuse with the ovum, but its skull has been lost, some part of the skull. Is that right? So what is really there? That because this part gradually fuses and multiple fusions form and then pores form, and then it is so. This is the price you pay. Not only you have to get your head washed, but you have to get a piece of a head lost. Is that right? Why you feel so happy with this? But this is what is really happening at cellular level. Right. Maybe no, sure, this happens like this. Right. Now listen. Now the sperm which reach first of now multiple sperm will touch here. I told you out of millions, hundreds will reach here. And multiple may be trying to go through acrosome reaction and trying to enter. As soon as, let's suppose there's one sperm here, there's another sperm which is here, another sperm which is embedded here. But let's suppose this is our friend, the lucky guy. It is the first one to touch the membrane of what? Ovum. Is that right? Of course, the fusion between the oocyte membrane and the sperm cell membrane, right? Remember, this is not the cell membrane or plasma membrane of the sperm from its head which fuses. No, head membrane has been lost. The real and actual fusion is between the posterior region of the sperm, because it has lost its part of the head, and the membrane of the ovum. Is that right? So you don't tell anyone that these membranes are really, this is the ovum. So this part of the membrane does not fuse with the ovum. It is this part of the membrane and this part of the membrane, because this part of the membrane is lost in acrosome reaction. Is that right? Now, as soon as, okay, let me make the sperm now, it is successful, it should be made with more respect. Right? And suppose this is the, what is this, nucleus of the sperm having how many chromosomes? 23 chromosomes, right? Rather we should say 22 plus X or 22 plus Y, depending upon, right? Now, anyway, 23 chromosomes, and this is the substance of the what? Sperm. And blue is the membrane, cell membrane of the sperm. Am I right? Now, the ultimate action will come. As soon as, listen, real things will start happening now. Conditioning has been done, capacitation has been done, acrosomal reaction has been done. I have told you that acrosomal reaction is induced by zona. Acrosomal reaction occur in the head of sperm, but it is stimulated by zona, which is the part covering of the ovum. Is that right? Now, again, if we talk about phases of fertilization, one phase was it had to pass through corona radiata layer. Only capacitated sperm passed through that. Am I clear? Second was the penetration of zona pellucida. We have talked about the penetration of zona pellucida is by acrosome reaction, and this reaction occurs specially. There are special zona pellucida protein number 3. We just call it ZP3. This ZP3 is specially stimulated. As soon as this touches the zona pellucida protein number 3, it undergoes acrosomal reaction. Is that right? ZP3 has multiple function. ZP3 also help the sperm to, yes, bind with the zona pellucida, and also help the sperm to undergo acrosome reaction. Is that right? Now, one thing is there, as soon as this membrane, sperm membrane will touch the ovum membrane, dramatic events will occur. What was those dramatic events? Actually, in the zona pellucida, there are multiple proteins. These are multiple species specific proteins. In the zona pellucida, there are multiple species specific receptor sites for the attachment of sperm. As soon as the first sperm membrane touch the, what is this? Ovum membrane. Ovum, again a very feminine behavior. It is having a lot of lysosomal enzymes in cortical granules. These are called cortical granules. As soon as there is a magic touch of the sperm, this cortical granules will move to the surface of ovum, and ovum will release lysosomal enzymes. And these lysosomal enzymes will bring changes in zona pellucida, and zona pellucida become impermeable to other remaining sperms. As I told you the last example, that analogy is that there is a female, she is sitting in a palace with 1,000 windows, and she is just waiting from some window someone should jump in. They don't tell it, but anyway. Now, the beauty is this, as soon as one man jumps from one window, the first thing she does is shut down all other windows. She doesn't want many men there. You understand it? Ovum has a similar very feminine behavior. It does not want many sperm to come in. That will create real mess and havoc. Polyspermy and all the genetic number of chromosomes will be disturbed. So, as soon as the first sperm's membrane touch the ovum, ovum undergoes cortical reaction. In cortical reaction, it releases cortical granules, and they release enzymes, and these lysosomal enzymes produce changes in the zona pellucida, and this reaction in the zona pellucida is called zona reaction. So cortical reaction lead to zona reaction. Is that right? During the zona reaction, zona pellucida alters and it inactivate species specific receptors. What are these species specific receptors? Anyone who knows why they are there?
Species specific.
Yeah.
Species specific.
Yeah. Why they are species specific?
You don't wanna mix a primate with a human or.
Yeah, you are right. You are intelligent enough to figure out that he says that these special receptor site allow to the human ovum, right? Allow only the human sperm to touch there and get attached there. Sperm from other sources other than the human males, right? They will not be able to penetrate through the zona pellucida. Of course, that's the good news. But what I was telling, that as soon as membrane touch sperm and this membrane touch cortical reaction occur, lysosomal enzymes come out into zona pellucida and then zona reaction occur and zona pellucida become impermeable. It undergoes structural and functional changes and it becomes impermeable to remaining other sperms, and especially it inactivates its species specific and receptor sites. Clear? Now that is why that in a fertilized egg, right, they find that there may be many sperms which are embedded in the zona pellucida, but only one really goes in. So magic touch has been done. The first one who comes touches the membrane of the female cell and all other windows or receptor sites are inactivated. Now what the sperm will do? Who will tell me? I'm going to see who is really intelligent. What sperm will do? Yeah? No, I'm saying, right, membrane of the sperm touch the membrane of the ovum, cortical reaction occurs, zona reaction occurs. Now it's a private matter between this particular sperm, the lucky guy, and the ovum. What is the next step? What sperm will do? Yes?
Probably doesn't need the tail anymore.
It doesn't need the tail anymore. It is very, very, you can say, disloyal to its tail or doesn't like its tail. It reminds him of the dogs or something like that. Yeah. No, look, he's clever. He says he will ask me question. No, but you have to answer first.
Okay.
That what sperm will do now? Yes, anyone? No. First of all, it will undress. It will leave its membrane out. It is sitting in the window, closing all the windows, undress there. Leave the dress out and then jump in. This is very important. Yeah, it is really happening like that. Right? I think this was some simple IQ question. That what sperm should do now? It has reached there. Their membranes are touching. Right? It will leave its membrane out. It will never take its membrane. It will just jump in the cytoplasm of the ovum only personally. I mean, look here. The sperm has come in, right, with its tail. And here is 23 its chromosome. Right? Is that right? And this blue membrane is left out. Usually, when we study the ovum, fertilized ovum, we find that membrane of the fertilizing sperm is just dangling with the membrane of the ovum outside. It's just like that. You find there's a window and some male dress hanging out. Right? The male is not there. Probably has jumped in. Right? So this is something like that which happens there. Is that right? There's an advantage of this thing. Ovum does not need to handle the membrane of the male sperm, isn't it? Now, ovum has to deal with the sperm only. Right? What it will do? I think somehow ovum does not like the tail. So first of all, its tail undergo degeneration. Is that right? What really ovum needed from the sperm was genetic material. Because ovum was having just 23 chromosomes. Now, one thing before really I go into further detail, let's talk about ovum nucleus. In the ovum nucleus, there were 23 chromosomes, but because this was secondary ovum, in secondary ovum, there are 23 chromosomes, but every chromosome is doublet. For example, this is chromosome number 1, maternal, chromosome number, okay, I will make, look, here, this is chromosome of ovum, and this is chromosome number of the sperm. Is that right? Now, this point is very important to understand. Right? Now, when chromosome come, when sperm come, every chromosome is only one. For example, chromosome number 1, only one copy. Chromosome number 5, only one copy. And in sex chromosome, it will have only X or Y. But by the time sperm touches it and trying to enter in, she is really not fully ready. Every chromosome is a double copy. So do you think it should be like that? No. So what we can say that sperm has 23 chromosomes and every chromosome has single DNA copy, not double. Secondary ovum has also 23 chromosomes but double copies. So what the ovum has to do? Immediately it will complete its second meiotic division. How? That half of the this chromosome will split in the center, and if this was chromosome number 1, one copy will go to one side, other copy will go to other side. From every chromosome, one copy will be kept with all the cytoplasm, and other copies will be thrown out as a small cell which is called polar body. So polar body is just a means to waste the unwanted genetic material. Are you understanding? So what this ovum will do after the magic touch of chromosome? It has to do cortical reaction and induce the zonal reaction so no one else come. Plus it should rapidly complete its second meiotic division and produce a polar body which will be later found somewhere here. Now of course, during the polar body formation process or during the completion of the second meiotic division, every doublet of the chromosome they separate. One group of chromosome get all the cytoplasm along with the nucleus, and other group of chromosome don't get cytoplasm much. That unfortunate one which does not get any cytoplasm, that is called polar body, because that is pushed on one pole of the whole zona pellucida, and remaining the ovum is called definitive ovum. So this is the process of fertilization which converts the secondary ovum into definitive ovum. Am I clear? Is that right? No problem? Now, so what is happening? Another thing I did not mention, but I think I should mention. If this is the ovum and this was the sperm, before really sperm enters, their membrane surface molecules lock in. What does it remind you? Before really it enters in, some molecules on the, what is this? Membrane of ovum, and some molecules on the membrane of sperm, they lock with each other like this, so that the first one which touches does not fall away. What does it remind you? I think nasty things coming to your mind. No, I'm not talking about that. This should remind you some important molecules. The molecule here is called integrins. Integrins. You know, female wants to have integration. Integrins. Right? Sperm wants to go in, but always behaving in some funny ways and displaying disintegrins. Disintegrins. So molecules on the ovum surface are integrins and molecules on the sperm surface are disintegrin. When I told you that they come and touch with each other, actually this is touching mechanism is by the interaction between the integrins and the disintegrins. That really locks the membranes of the two cells together, so that this sperm which has traveled such a long travel should not, after reaching to the ultimate place, it does not fall away. Is that right? So this is how they are held together, right? Now, if someone asks you that once the sperm touches here and it is entering in, how the egg responds, you should give 3 answers. What is the response of the egg to the arrival of the and magic touch of the sperm? There are 3 answers. Number one was cortical and zonal reaction. Cortical reaction we have discussed in detail already. You know, the first purpose should be what? All remaining windows should be shut down. Right? So this is one. Number 2 is completion of second meiotic division. Completion of second meiotic division. Again, what I'm asking, that when the sperm really touches the ovum have to do certain things in the honor of arrival of sperm. Number 1, it undergoes cortical reaction and bring alteration zona pellucida and prevent the entry of other sperms. Number 2, ovum in hurry complete its second meiotic division, throws out one polar body, and now secondary ovum is converted into definitive ovum. And number 3, that ovum does its metabolic activation. Actually, ovum without sperms, they are in a good mood or bad mood. They should be high metabolically, too active or less active. You think ovum without the sperm should be more active? I think there's a common sense, isn't it? That ovum, and if sperm never comes, ovum is less metabolically active. When the magic touch of the sperm is there, ovum become metabolically very active, because it has to do many metabolic reactions and it has to divide into 2-cell stage, 4-cell stage, morula, blastocyst, bilaminar disc, trilaminar disc, embryo, fetus, right? And maybe eventually it become Luis Alberto. So, ovum has to become very metabolically active. That is also because if sperm never comes, ovum does not become truly metabolically active, and due to that reason, within 24 to 48 hours, it dies. But fertilized ovum lives for long time. Is that right? So, 3 things happening to the ovum. Number 1, it is doing the cortical reaction on the arrival of the sperm. Cortical reaction. Number 2, it hastily completed its second meiotic division and convert from secondary ovum to definitive ovum. And number 3, it metabolically activates itself for the very good bright future which is coming there, right? Of course, metabolic activation factor is provided by the sperm. Don't underestimate them, even though they are small, right? Meanwhile, when ovum is doing its functions, what happens to sperm? I've told you that it has left its dress behind its membrane and it has jumped in naked. Then its tail is cut off, it degenerates. And then its cytoplasm also disappears. Now, what is this? This is nucleus from the ovum. Nucleus from the sperm. This is the nucleus from the sperm. And meanwhile, once polar body has been thrown out, this is a nucleus from ovum. Is that right? Let me make it more sensible that this should be blue. This is nucleus of the sperm, and this red one is the nucleus of ovum. 23 chromosomes are present over here, and 23 chromosomes are present over here. Now what this is, before they really touch each other, now look at the beauty. It's not only male and female touching and leading to fertilization. The sperm and the ovum touching each other. Not only that, even these nuclei will do special preparation to greet each other. Now this will do very special preparation. Many of the enzymes inside it become active. And this will also become super active. Then they will meet each other. So what they will do, both of them? They will double their DNA. This will double its DNA, this will also double its DNA, genetic material. It was 23 chromosomes. It will make from 23 single chromosome to 23 doublet. For example, again, it's a very important concept to understand. Look, this is a female chromosome, right? And this is in the nucleus of the definitive ovum. And here is a male chromosome, right? Is that clear? Now, this is maternal chromosome number 1, maternal chromosome number 1, and suppose this is paternal chromosome number 1. Baby should get both, 1 copy of paternal and 1 copy from maternal. But you know, they are thinking of the future. This cell, now this is zygote. This cell will soon divide into 1 cell or 2 cells? 2 cells. So, what really happens? This is the nucleus brought by the sperm, and this is the nucleus present by the definitive ovum. Now, before the nuclei greet each other, male nucleus and the female nucleus as they are called, this will double its genetic material. It's so beautiful. I will teach you in lectures of molecular biology that its DNA start uncoiling and enzymes jump in. From this DNA they will make on DNA. On 1 chromosome maybe 3000 genes. So exactly 3000 genes are copied here. In the same way, this also copies itself. Now tell me, how many maternal chromosomes are present here? And how many paternal chromosomes are present here? Don't tell me two. Chromosomes are not counted by their DNA number. Chromosomes are counted by their centromeres. This is still one chromosome with double DNA. And this is also one chromosome with double DNA. Is that right? So these will become 23 chromosomes with double genetic material. So 23 chromosome, which when it came, it was 23 with one N number. Now it becomes 23 with 2N. And this becomes 23 with 2N. Yes, what is your question?
If it has to copy it, then why does it bother doing the polar body thing if it needs it anyway?
Oh my God, she's asking that if it was supposed to do the copy, why it bother with the polar body thing? Actually, this is a basic principle that before the mitotic division, this is the basic thing we learn in mitosis, that before the mitosis, genetic material has to be doubled. Our molecular machinery, enzymes are pre-programmed for this thing. And if it was not bringing the polar body out, from 2 it will make 4. Right? So this is a purpose in nature. Am I really clear to everyone? So this is, now there are 23 chromosomes with double copy of genetic material, 23 chromosomes with double copy. Now they are ready for the next generation. Maternal chromosome and paternal chromosome. What they will do? As this process, when they are making their, when they are making their genetic material double, this process is called growth of the nuclei. So when this is called male pronucleus and this is called female pronucleus. So we say that in the fertilized ovum, male pronucleus and female pronucleus go under the growth and maturation. What they are doing then, they are doubling their genetic material. And after that what happens? They will come together to each other. And you know, whole purpose of all this thing was to combine the maternal genetic material with paternal genetic material and make a new person in the world with a unique combination. Now, when they move to each other, here are the paternal chromosome, here are the maternal chromosome, only barriers are these membranes. So as these two pronuclei, male and female pronuclei, meet with each other, they immediately, no, they touch and run away. They are rubber balls? No. Again, remember important things in life. Both of them will undress. Both of their envelopes will dissolve, melt away, and these chromosomes will become happily out. But first they have to touch each other. You know, magic touch means a lot in biological world. As soon as they touch each other, these 23 chromosomes and these 23 chromosomes, they are too desperate to meet each other to make a new human being, so the membranes dissolve. But this is 23 with double copy. I mean, it is like this and like this. And this is 23 also with double copy. But I am just showing, suppose chromosome number 1 maternal and this is chromosome number 1 paternal. Is that right? Now this is the intermingling of maternal and paternal chromosome, which was a part of the fertilization process. Fertilization process is continuing still. Is that right? So both membranes disappear, chromosomes, maternal and paternal, intermingle with each other, and after having initial greetings, they decide to depart to make two cells. So what will happen? Spindle will be formed in, what will be made? What will be made? Spindle will be, metaphase will come, and on the spindle, maternal and paternal chromosomes will be decorated first. For example, this was which one? Paternal, chromosome number 1, and what is this? Maternal, chromosome number 1. Now what will happen? This chromosome will longitudinally break at its centromere, and one copy will move to this end, other copy will move to the other end. And in the same way, maternal, one copy will go to one end, other copy will go to the other end, and deep furrow will develop on these side. Cell is trying to go into cleavage. So soon you will find that one copy of chromosome 1 from mother come here, one copy of chromosome 1 from maternal side come here, one copy of What is this? Paternal come here. And when this deep furrow will go, how many chromosome number 1 this cell has? 1 paternal and 1 maternal. 23 paternal, 23 maternal, 46. Am I really clear? And 46 in other cells also. Right? No problem up to this? One thing, initially this chromosome was doublet. Suppose this was doublet. Then it will split here. Then this will move on this side. This will move on this side. When they are moving, what are they called? 1 chromosome or 2 chromosomes? Then they are called chromatids. Have you heard of the term chromatids? Right? Now this is the movement of the chromatids. And when they reach up to the end and again they dress up themselves, new nuclear membrane start, then they are called chromosomes. And then every time they will double themselves, again divide. Double themselves, again divide. Millions of the cells will be made. But every cell will have the same genetic material as the fertilized ovum had initially. Is that clear? No problem up to this? Now, again, just to sum up, what are the main results of fertilization? What are the main results of fertilization? Number 1, establishment of diploid number, because sperm had haploid number, 23 chromosome, and ovum eventually has 23 chromosome. But after fertilization, male and female pronuclei form, then eventually they fuse, genetic material intermingles, and cell from haploid has become diploid. Is that right? So establishment of diploid number of chromosome. Number 2, establishment of chromosomal sex of the new individual. Because all the ova have X chromosome, but half of the sperm have X chromosome and half of the sperms have Y chromosome. So actually at so early stage, at chromosomal level, sex has been determined. If ovum brought the X chromosome, the maternal X plus paternal X make a female baby, female fetus. But if sperm has brought Y chromosome and ovum was having X, then what happens? X and Y, male baby. Is that right? So another result of the fertilization is that at that very moment, the moment fertilization has been done, right, chromosomal sex of the product of conception is established. Genitalia will develop, of course, later, right? Third main result of fertilization is metabolic activation of the ovum and initiation of cleavage, so that one cell go into 2 cell. Because if sperm is not there, do you think ovum alone will go into 2 cell state? No, right? So third result of fertilization is initiation of cleavage. Is that right? Any question up to this? So here we complete our lecture of fertilization. Next lecture we'll talk about how this unicellular embryo, right, fertilized ovum or zygote, right, how zygote convert into 2 cell stage and 4 cell stage and then morula and then blastocyst and so forth. That will be discussed in coming lectures. Class dismissed.